All right, sounds good. Okay, guys, today is chapter 6. I'm Calvin. If you guys hadn't seen the chapter 1 presentation, I did that one, but I'm Calvin. I'm now a second-year medical student coming up this August, and survived my first year, thank goodness. And today we're going to be taking a look at chapter 6, which is entitled Somatic Sensation, Trigeminal and the Viscerosensory Systems. And all right. Now the outline of the talk is presented here. We're going to start with a clinical case. It poses some questions that without knowing about the chapter, supposing you have no prior knowledge, you wouldn't be able to answer. So we'll look at the questions and then deal with them later when I do go over that material. Then we'll take a look at the cranial nerves and the nuclei, some of the differences between them and the limb and the trunk nerves. There are 7 functional categories of cranial nerves. And then finally, we'll take a look at how the nuclei organize themselves and develop into distinct columns in the brainstem. Then we'll move on to functional anatomy, looking at facial mechanosensation, pain sensation, this really important structure called the caudal solitary nucleus. And then we'll go into more regional anatomy, looking at the innervation of the face, mucous membranes of the head, spinal cord. Another important nucleus, in addition to the caudal solitary, the spinal trigeminal nucleus. Another one, the mesencephalic trigeminal nucleus. More nuclei, and then we'll take a look at some practice questions if we have time. So we're starting with this clinical case. Here we go. We have a 69-year-old male presenting with sudden onset vertigo and difficulty walking. Now he has facial pain and thermal sensation absent, the gray tint on his left side of his face here, and then the arm, trunk, and leg on his right side. Okay, so he's lost pain and thermosensation in those grayed out areas here. And then mechanosensation spared on the face, limbs, and trunk. So it's only the pain and thermal sensation. So he can feel things touching him, but not pain nor heat. Jaw and limb proprioception spared, so he knows where his limbs are. And then finally, on MRI, we have a wedged shaped lesion dorsolaterally on the left side, showing up really quite small, but it is right here in that region there. Okay, so questions that we'll look at for the end of the talk is what artery supplied the infarcted region of the medulla, which is up here in the MRI. Oops. And then secondly, why is there this ipsilateral, contralateral sensation loss? All right, so keep that in the back of your mind, but now we're gonna move on to the cranial nerves and nuclei. So to start, here is Martin's diagram of the 12 cranial nerves: the olfactory, optic, oculomotor, trochlear, trigeminal, abducens, facial, vestibulocochlear, glossopharyngeal, vagus, spinal, accessory, and hypoglossal. So now to go into these a bit more, these blue ones, the olfactory and the optic nerves are purely sensory. The olfactory nerve, which mediates the sense of smell, directly enters the cerebral hemisphere. The optic nerve for vision enters the thalamus. The other 10 cranial nerves, so those are 1 and 2, the other 10 enter and leave the brainstem on this picture here. So now taking a look at the 2 red ones, the oculomotor and the trochlear nerves, these are both motor nerves and they exit from the midbrain here. Both innervate muscles that move the eyes. And the trochlear nerve is further distinguished as the only cranial nerve found on the dorsal brain surface. So now looking at these 4 green boxed nerves, the trigeminal nerve is located at the middle of the pons. It's a mixed nerve, which means that it has both sensory and motor roots. The sensory root provides the somatic sensory innervation of the facial skin and mucous membranes of parts of the oral and nasal cavities and the teeth. And then the motor root contains axons that innervate jaw muscles. The remaining 3 pontine cranial nerves down here, whoops, sorry. The remaining 3 pontine cranial nerves here, these green ones, are found at the pontomedullary junction. The abducens is a motor nerve that, like the oculomotor and trochlear nerve, innervates eye muscles. The facial nerve here is a mixed nerve and has separate motor and sensory roots. The motor root innervates the facial muscles that determine our expressions, whereas the sensory root primarily innervates taste buds and mediates taste. The vestibulocochlear nerve here is a sensory nerve that has 2 separate components. The vestibular component innervates the semicircular canals, sacculae, and utricle that mediates balance, whereas the cochlear component here innervates the organ of Corti and serves hearing. Now, finally, the 4 orange nerves. The medulla has 4 cranial nerves here in orange, each of which contains numerous roots that leave different rostrocaudal locations. So starting with the glossopharyngeal nerve, cranial nerve 9, it's a mixed nerve, and its major functions are to provide the sensory innervation of the pharynx and to innervate taste buds on the posterior 1/3 of the tongue. The vagus nerve is a mixed nerve and has myriad sensory and motor functions that include somatic and visceral sensation, innervating the pharyngeal muscles and much of the visceral or internal organs autonomic innervation. The spinal accessory and hypoglossal nerves, cranial nerves 11 and 12, subserve motor functions, innervating the neck and tongue muscles, respectively. So that's a lot. I've here summarized or taken a chart that summarized each of those functions into a nice table. So these nerves are really important. It also gives us a nice mnemonic here if you'd like to remember them that way. And then also a different view of the locations of where these cranial nerves are emerging. Table 6.1 in the text, which was a little bit cut off, so I couldn't get it in to my presentation, but it mentions the foramina, or the openings in the bone, where the cranial nerves pass through. So I just want to mention those briefly. The olfactory nerve, cranial nerve 1, passes through this cribiform plate, the optic nerve, through the optic canal here. So you can kind of watch these. This chart works like this in this horizontal direction here. The superior orbital fissure has 4 cranial nerves, the oculomotor, trochlear, abducens, and the ophthalmic division, V1, of the trigeminal nerve. To point out the trigeminal nerve, V1 goes through the superior orbital fissure here, V2 through the foramen rotundum, and the V3 through the foramen ovale. There's this nice mnemonic here, cranial nerves. Cranial nerve 5 exits V1, standing room only, SRO, and then superior rotundum ovale. So it's a pretty cool way to do it. I won't go through the other ones, but this is one thing that was in the text that I wasn't able to get directly from the text. Now, innervating, when looking at the innervation of that comes from the cranium and then the limbs and the trunk, there are 3 similarities in those types of innervations. The first being that the distal portion of the axon of sooty unipolar primary sensory neurons is sensitive to stimulus energy, so that's both cranial and limb and trunk innervation. The cell bodies of these primary sensory neurons are located in the peripheral ganglia, and then the proximal portion of the axon projects into the CNS to synapse on neurons in the medulla and the pons. So now we're going to look at the differences. We're going to look at what's unique about cranial nerves and how they pick up information and then deliver them. So starting down here with the purple, teal, and orange boxes, for the senses of taste, vision, hearing, and balance, a separate receptor cell, here I boxed in gray, transduces stimulus energy. So these are specialized cells that are particularly designed to pick up on taste, hearing, balance, and vision in these 4 examples, or 3 examples. So it's these receptor cells that activate synaptically the primary sensory neurons depicted here, which transmits information encoded in the form of an action potential to the central nervous system upstream. Now looking at the blue and the green boxes, blue and green here, for the spinal and trigeminal somatic sensations, which are these 2 boxes, the distal ending of the primary sensory neuron is the sensory receptor for all but one receptor. And then the primary sensory neurons and cranial nerves have either a pseudo-unipolar or a bipolar morphology depicted there. Now the red box, jaw proprioception, there are stretch receptors in jaw muscles which signal jaw muscle length and thus mediate jaw proprioception. They're pseudo-unipolar primary sensory neurons, but their cell bodies are located within the central nervous system, not the peripheral ganglia. Another thing is that most neural crest cells migrate peripherally and give rise to neurons whose cell bodies lie outside the central nervous system, excuse me. However, the primary sensory neurons that mediate jaw proprioception, here in red, derive from a special group of neural crest cells that do not migrate from the central nervous system to the periphery. So they, they do not migrate from the central nervous system to the periphery, these jaw proprioception cells. Okay, 7 functional categories of cranial nerves. Now, there are 4 categories that are similar to the spinal nerves. First, there are somatic sensory fibers, touch, pain, itch, temperature, jaw and limb proprioception. Then we have viscerosensory fibers mediating the visceral sensation, chemoreception from body organs and help regulate blood pressure. Somatic skeletal motor fibers, these are the axons of motor neurons that innervate striated muscle that develop from the somites. And then 4th, similar function, is the visceral autonomic motor fibers. These are the axons of autonomic preganglionic neurons. Now there are 3 additional categories for the cranial nerves. First, there are axons that innervate the eyes that subserve vision. And then there are axons that innervate the inner ear that mediate hearing and balance. Next, there are fibers innervating taste buds and the olfactory mucosa mediating taste and smell. And then finally, branchiomeric skeletal motor fibers. These are the axons of motor neurons that innervate striated muscle that develop from the branchial arches. Okay, so here we're taking a look at how cranial nerves develop, and then on the next couple slides, we'll see their organization into columns, distinct columns within the brainstem. So here on the left, the spinal cord in its development has a segmental organization, and this organization arises early on in development. The developing pons and medulla have 8 segments up here in red, termed rhombomeres. It's a blue box, but on the picture they're red. These provide sensory and motor innervation from most of the head to the cranial nerve peripheral projections. So, in comparing cranial nerves again to spinal nerves, there are 3 important differences between cranial nerves and spinal nerves as far as their development. First, the sensory and motor nuclear columns in the medulla and the pons are aligned roughly from the lateral surface down here laterally from, excuse me, roughly from the lateral surface of the midline, rather than being oriented on the dorsal-ventral axis as in the spinal cord. I see. So here, this looks more like, let me start with this B figure. These are sections of the brainstem through fetal development, early, later, and later. Here, this is a dorso-ventral organization of the brainstem early on in its development. Here's the brainstem later on in development, and you can see it transitioning from this dorso-ventral organization, like the spinal cord, even in adulthood. Through time, there's dorsal expansion, and then there's a more lateral to midline organization, sort of across horizontally like this. That's the point it's trying to make. So instead of the spinal cord being and remaining in a dorsoventral organization, the brainstem spreads out and opens up sort of to have this lateral to midline organization. The second difference is that the brainstem integrative nuclei derived from developing neurons of sensory and motor plates in the ventricular floor, and the immature neurons that give rise to these structures in the brainstem migrate in teal here, the migrating neurons, to their destinations in dorsal and ventral regions. That only happens in the brainstem, not really in the spinal cord. Now, here, the third difference relates to these columns that I've mentioned a couple of times, which is how the cranial nerves are organized in the brainstem. So, and also the greater diversity of cranial nerve nuclei compared to the spinal nerve nuclei. So because there are 7 functional categories of cranial nerves, there are also 7 categories of cranial nerve nuclei. And nuclei from each of these categories form discontinuous columns that extend rostrocaudally throughout the brainstem. First point here in blue and red is that the sensory columns in blue, balance and hearing, somatic afferent, visceral afferent, and taste, these sensory columns are located laterally to the motor columns in red. So all these reds and oranges are motor, and these blues are sensory. The somatic sensory hearing and balance system, balance columns in green tend to be lateral to the viscerosensory and taste columns in purple, boxed in purple. So you can kind of see these two as located laterally to this dark blue, which are these visceral afferents. Then the somatic skeletal motor neurons in gray, boxed in gray, is medial to the autonomic motor column, here again in teal. And then the branchiomeric motor column in orange contains the neurons that are located in the region of the reticular formation up here. So there's, this loops around here to this darker orange. Whoops. Keep doing that. Now, finally, the sulcus limitans is a shallow groove that separates the sensory and motor columns during development and remains as a landmark on the floor of the 4th ventricle in the adult brain.
I just want to add, when you become a resident, you're going to have to draw these cross sections. And so thinking about it this way, just in general, where things are can really help you kind of think through at each segmental level of the brainstem, you know, where each column and where each nucleus is. Obviously, it's going to vary. It's not the same throughout the entire neuroaxis as the left, as panel A shows. But it's a very helpful way to think about it.
Yeah, thanks. So these columns are located in this nice organization relative to each other, and Shane just pointed out it's helpful to know their relative location to each other. Okay. Now, because cranial nerve nuclei that serve similar functions are aligned in the same rostrocaudal columns, knowledge of these locations of these columns aids in the understanding of their functions. So really jumping off Shane's point, if you know their location, you know where they're located near a group of certain cranial nerve nuclei will give you a clue about what their function is, because like I just said, cranial nerves that have similar functions are located close together. Now this figure just gives you another view of the longitudinal organization of the cell columns forming cranial nerve nuclei in the mature, the adult brainstem. So those colors, those reds, those blues, and each individual cranial nerve is here labeled. Okay, moving on to functional anatomy next. So first, the trigeminal nerve. It's a big part of our talk, the trigeminal nerve here, the trigeminal system. So it innervates most of the head and the oral cavity. Three other nerves, cranial nerves, facial 7, glossopharyngeal 9, and vagus 10, contain sensory fibers that mediate taste. And then they also, on the skin, innervate small areas of the ear, skin around the external ear, then the mucus, some mucous membranes, and then the internal organs, especially the vagus nerve, is really important for respiration and blood pressure. Now, I want to talk about sensory fibers of the skin and oral mucosa, particularly. So sensory fibers that innervate surface skin and oral mucosa project into central trigeminal nuclei. There are two of them here, boxed in blue, the mesencephalic trigeminal nucleus and the main trigeminal nucleus. I'm going to touch on each of these in greater detail later. The second nucleus, the solitary nucleus here in red, sensory fibers that innervate the mucous membranes of the pharynx and larynx, and other visceral structures project into the caudal portion, the lower portion, of the solitary nucleus. Again, these are just an overview of what these two nuclei are receiving inputs from. Now I want to draw a distinction in
contributions. Those afferents are going to be from different cranial nerves. So that's the tie-in to what we were talking about earlier when you were going through all the different cranial nerves. The lower cranial nerves, like 9, 10, will go into the solitary nucleus. It's the same sensory information, but just of the pharynx and the larynx. Mm-hmm. The stuff from the face and the oral mucosa higher up in the aerodigestive tract is going to go to the trigeminal system, which I'm sure you're going to talk about.
Yes, I think there's more to come later, but I'm not sure I pointed out the exact cranial nerves that go to each of these structures. It was more descriptive, like I have. There's more detail, but like I said, I don't think I have the exact cranial nerves. So that's a good thing to point out. That particular cranial nerves go to particular nuclei in the trigeminal system. So now, distinguishing between large and small diameter fibers. Large ones mediating mechanosensation, these terminate in the main trigeminal sensory nucleus, number 2 down here. Then small diameter fibers, these mediate pain, temperature, itch, and itch. These mainly travel in the spinal trigeminal tract, which I'll point out later, and then they terminate in the spinal trigeminal nucleus, which has these 3 components that you can read here, and I will touch on those later. So just think about these different functions, mechanosensation, pain, temperature, and itch, ending up, these afferents are ending up in different parts, different nuclei in the brainstem. Now, this sort of sets the stage. These two nuclei here, these 2 different functions for each of these 2 nuclei sets the stage for 2 different trigeminal ascending sensory systems. They're anatomically and functionally distinct. The first one, A, I've called it A, has to do with cranial touch and dental mechanical senses. The second one, B, in red, has to do with cranial pain, temperature, and itch. So let's start with A. First, most neurons in the main trigeminal nucleus sectioned here in the pons in green, this main trigeminal sensory nucleus receives, these neurons receive mechanoreceptive information. Oh, I do have the cranial nerves from cranial nerves 5, 7, 9, and 10 in the gray box. First order, they synapse here in that nucleus and then ascend, which I have a bullet on here. These axons from this nucleus decussate in the pons. The ascending second order trigeminal fibers are termed the trigeminal lemniscus. This is sort of the pathway they take up, arising through the midbrain and then eventually into the thalamus, which I'll mention here. These second order fibers synapse in the ventral posterior medial nucleus of the thalamus in teal, VPMN here in the thalamus. And then the third order neurons I have here in Dunkin' Donuts colors, in orange. So these axons of third order thalamic neurons project via the posterior limb of the internal capsule, this tract here between the thalamus and the primary somatic sensory cortex, which actually I presented on in chapter 1, and that's located in the post-central gyrus. Okay, so that's the first pathway, A. Now we're going to take a look at B, cranial pain, temperature, and itch in red. Okay, the spinal trigeminal nucleus has a rostral caudal organization with 3 parts. I mentioned this a little bit before. First is the oral nucleus. Second, the interpolar. And then next, the most caudal, aptly named, is the caudal nucleus. These are depicted here in sections of this part of the brain here, 1, 2, 3. This structure, the spinal trigeminal nucleus, plays an essential role in facial and dental pain, temperature, sensation, and itch, like I mentioned. Now, moving along up, the major trigeminal pathway containing second order neurons from the spinal trigeminal nucleus is organized into the trigeminothalamic tract here in purple. So first order neurons, these are also coming from cranial, I know it's really small, I apologize, from 5, 7, 9, and 10. These sort of travel downwards and synapse in the, in green, the spinal trigeminal nucleus. Then they rise up through the trigeminothalamic tract. A lot of big words in this talk. And then they terminate, these second order axons terminate at the contralateral thalamus. So they do cross over. Notice where the first order neurons are coming from. And then on the contralateral thalamus is where they're ending up. And then just a small point that the one of 3 nuclei in the thalamus, the ventral posterior medial, ventral medial posterior, or the medial dorsal. My goodness. Okay, I pointed those out here in teal. Okay, so for now moving up to the third order, I've just chopped off the top part of the figure here. The ventral posterior medial nucleus of the thalamus projects to the primary somatic sensory cortex, similar to the previous ascending pathway A. Ventromedial posterior nucleus projects to the insular cortex, and then the medial dorsal nucleus projects to the anterior cingulate gyrus. So these, this sensory information here in pathway B, these have a more diverse set of final cortical areas in the top of the brain as their final destination. So that's a key distinction. But one thing I did want to point out is that both A and B, both of these 2 ascending pathways, do communicate with the ventral posteromedial nucleus. I just boxed the trigeminal pathway because that's what we're talking about. And this chart only says face sensation, but now we know that it's a bit more than that. Touch, dental mechanical senses, cranial pain, temperature, and itch. And then finally, I do like the mnemonic here for the ventral posteromedial nucleus. Makeup goes on the face, so we can kind of view these two ascending pathways as dealing with the head and face, touch, pain, and temperature and itch on the head and face. Okay, the caudal solitary nucleus. We can view it as the origin of the viscerosensory system. And the glossopharyngeal and vagal axons innervate the pharynx and larynx, the esophagus, the thoracic and abdominal viscera, and the peripheral blood pressure sensors. And these axons from cranial nerves 9 and 10 project to this caudal portion of the solitary nucleus, forming the origin of the viscerosensory system. From this nucleus, we're kind of tracking along up on these various different pathways. From this caudal solitary nucleus, there are local projections to the medulla and the pons that mediate blood pressure, really important, respiratory rate, and GI motility and secretions. So these are like lower brain functions, that local projections from this caudal nucleus. That's where it's communicating with sort of these low brain functions. Sensory information processed here helps initiate protective reflexes such as the laryngeal closure reflex, originating here from the caudal solitary nucleus. And then finally, descending projections to the spinal cord directly control portions of the autonomic nervous system. Okay, now we're taking a look at the full picture of the caudal solitary nucleus's ascending pathway, and I boxed the portion that we're looking at down in the brainstem. So in the medulla, here's the caudal solitary nucleus, and then further up in the medulla is the solitary tract. Now, here are cranial nerves 9 and 10, cranial nerves 9 and 10 mediating viscerosensation, enter the brainstem and collect into the solitary tract, moving down here. Then these axons terminate in the caudal solitary nucleus. Now, for conscious awareness of viscerosensory information, such as feeling nauseous, there is an ascending projection via the parabrachial nucleus here in green to a portion of the ventral posterior nucleus of the thalamus, which then projects on to the insular cortex, which I believe I have here, yep, to the insular cortex in teal, so that you can be consciously aware of that sensation. So just to read, these viscerosensory thalamic neurons, which are distinct from mechanosensitive and taste, project to the insular cortex. So that's sort of the final destination of information coming through the caudal sensory nucleus mediating viscerosensation. Okay, the final last big chunk of our talk is going to discuss regional anatomy of the trigeminal and viscerosensory systems. Now, to start, the trigeminal nerve is aptly named trigeminal, because it has 3 sensory roots. It has 3 roots, but as far as their sensory functions, the trigeminal nerve consists of these 3 sensory roots that innervate skin and mucous membranes of 3 different regions of the head, as depicted over here in this picture. In blue, the ophthalmic division, a pretty big region from the eyes extending backwards in this light yellow. The maxillary division, here in this lighter beige sort of thing here from the nose and lips. And then finally the mandibular is this dark yellow color here. One thing I do want to point out is that the cell bodies of the trigeminal sensory fibers that mediate cranial touch, pain, temperature, and itch are found in the trigeminal or semilunar ganglion. These are both peripheral sensory ganglion. But by contrast, the cell bodies of stretch receptors found in the jaw muscles, these are located in the CNS, in the mesencephalic trigeminal nucleus. This is that neural crest migration story. So it's a similar theme to that. And then a final point, the trigeminal nerve also innervates stretch receptors in the extraocular muscles. but the cell bodies of these fibers are located in the semilunar ganglion in their axon's course within the ophthalmic division of the trigeminal nerve. So three really important divisions of the trigeminal nerve, making for a very expansive set of functions that the trigeminal system mediates. Okay, now continuing on our task of determining what allows us to sense things on our cranial skin, also innervating portions of the skin and the head. Together, these three in purple. The intermediate nerve, which is a division of the facial nerve. These cell bodies are located in the geniculate ganglion. Geniculate ganglion. The glossopharyngeal nerve, cranial nerve 9. These cell bodies are in the superior glossopharyngeal ganglion. And then the vagus nerve, cranial nerve 10. These cell bodies are in the jugular ganglion. The reason I mention the ganglia are because a table in the text did point them out. It was, again, that table that was cut off, so I've included them here. They are in the text. The text had a discussion comparing and contrasting the glossopharyngeal and the vagus nerves, and I wanted to take a minute to touch on that. So starting with the glossopharyngeal nerve, it innervates the posterior 1/3 of the tongue, so the back 1/3, the pharynx, I don't know why I'm pointing, whatever, the pharynx, the nasal cavity and sinuses in the eustachian tube in the back of your ear. The vagus nerve, here in red, innervates the hypopharynx, the larynx, the esophagus, and the thoracic and abdominal viscera. Now, to look at their functions together, here in purple, both of them are really important for these three functions: swallowing, keeping airway clear of saliva and other liquids, and then finally, innervation of blood pressure receptors in the carotid sinus and the aortic arch. That is the baroreceptor reflex. So both the vagus and the glossopharyngeal nerves are really important for these three actions in the purple box. Okay, now we're taking a close look at the spinal trigeminal tract, a very important structure that extends from the spinal cord up through the lower parts of the brain. So starting with the blue boxes, the intermediate glossopharyngeal vagus and mandibular division of the trigeminal nerve are located dorsal. So this dark, sort of mustard yellow sliver here are the intermediate glossopharyngeal and vagus, and then also one next door neighbor is mandibular, this sort of lighter yellow, or more pure yellow. The ophthalmic division of the trigeminal nerve is located ventral here in red. So all the way ventral, this light yellow, here's the ophthalmic division where it arises or leads to, and then the maxillary is in between in green. So now taking a look, that was sort of the dorsoventral organization as these sort of distinct slices of this part of the brain. Now we're looking at the rostrocaudal organization, so up to down. The following lettered regions process somatosensory information, pain, itch, and temperature from the A, sort of up top, is the perioral region and nose. Two sections below, both B and C, are the posterior face and ear. And then finally, D is the arm, neck, and occiput. So you can view these rostrocaudal sections of this part of the upper spinal cord, caudal nucleus and interpolar nucleus, as subserving similar functions, processing somatosensory information, but from different portions of your body, sort of on a rostrocaudal axis. It sort of corresponds to where the information is coming from, but the type of information is the same.
I remember when I first learned this, I found it extremely confusing. Can you go back 2 slides, I think? One more. One more, there you go. So when you look at those dotted lines, A, B, C, and D, that correlates with what we were just seeing.
Oh, yes. Good
So
point.
it's like think of it like a matrix almost, okay? So, and the way to think about it is during evolutionary development, you could imagine if this was like a quadruped, like a dog, that the tip of the nose is the most anterior part, and the dermatomes, right, go backwards from there. So think of A, B, C, and D as the dermatomes of the head and neck, right? Because they basically are completely in line as you continue to go down the neural axis into the spinal cord with the segmental organization of the spinal cord, right? Whereas the exact location and nerve through which that information in that dermatome can come from mixed parts of the trigeminal nerve. It can come from either V1, V2, or V3. So that's the rostrocaudal dermatomal type organization within the brainstem. And then what you see in yellow and in the dark gray color for the lower cranial nerve nuclei, that's the actual specific nerves that transmits them. But it's kind of a matrix, and each of those is seen throughout the entire length of the brainstem because they all have different contributions to each dermatome.
Thanks, Shane. Yeah, it's kind of a tangled web, but I do like
It is.
I like the matrix idea where you can kind of view these divisions of the spinal cord and lower brain here, A, B, C, D, but then the exact nerves that this information is traveling through is based on these yellow colors. Yeah, I like that. I think it would take more pondering for me to actually understand it completely, but on the surface level, no pun intended, this is what the text presented. Okay, here's that. We did this. We did this. Okay, here we go. Continuing the spinal trigeminal tract, now re-looking at the caudal solitary nucleus. So axons in the spinal trigeminal tract, nicely named synapson neurons in the spinal trigeminal nucleus. So tract in blue to nucleus in red. And these are two different rostrocaudal sections of this lower part of the brainstem here, A and B. Then the caudal solitary nucleus and tract are located more centrally here in green. These are located in the caudal medulla. Okay, so there are three trigeminal nuclei that I've labeled in green, red, and blue. Starting with green, the spinal trigeminal nucleus. It mediates facial pain, temperature, and itch. The main trigeminal nucleus mediates facial touch, oral mechanical sensation here in red. And then in blue, the mesencephalic trigeminal nucleus mediates jaw perception, or proprioception, excuse me, here up here, okay. Now, here we go. A bit of vasculature for you. Posterior, inferior, cerebellar artery. And because that's a mouthful, we just say PICA. Here it is labeled on this figure. It provides the arterial supply to the dorsolateral, here in green, portion of the medulla. It's an end artery, which means it has very little collateral flow from other vessels in the territory it serves. It's kind of isolated. Therefore, the dorsolateral portion of the medulla in green becomes infarcted, hint, hint, when PICA is occluded. And the medial region of the medulla here is spared because there's collateral blood flow from the contralateral vertebral artery and the anterior spinal artery. So the occlusion of PICA produces a complex set of sensory motor deficits termed lateral medullary syndrome or Wallenberg syndrome. And I'm not going to go through this. It's another table describing the posterior inferior cerebellar artery, sort of some of the other symptoms. And then it does mention lateromedullary. This is more for your reference, but I want to get straight to our clinical correlate from the beginning. We had a man with sudden onset vertigo and difficulty walking. Here's the key. He's lost pain and thermal sensation on his face on the left side, the ipsilateral side, and then his arm, trunk, and leg on the contralateral side. Remember that the MRI lesion showed the lesion, or the MRI showed a lesion on the left here. So the question then, I've given it away, which artery is it? Pica. Now we need to explain why pain is lost ipsilaterally on the face and then contralaterally on the limbs. And to do that, we're going to take a look at this picture that we just looked at. So pica occlusion, it's going to cause damage to the spinal trigeminal tract and nucleus at the mid-medulla. This damage occurs before decussation. So notice here in blue, the spinal trigeminal tract and nucleus, these nerves have not yet decussated, or we're arising directly from it, and it's going right up to the thalamus there. So that's why we lose ipsilateral face nociception and thermosensation. Contrast that with pica occlusion also causing damage to the ascending anterolateral pathway. This damage occurs after decussation, which comes from, which then crosses over before it gets to this mid-medullary area. So that's why loss of contralateral limb trunk nociception and thermosensation occurs. So it's about, the key is where decussation occurs. Here it's before, and with the anterolateral pathway, it's after.
And Calvin, I just want to, just for you, when you rotate with us, we usually call that pica, not pica, okay?
Oh, okay.
Yeah, that's usually the way we pronounce it. Some people might give you a hard time for that. So I just want to...
Pica. Thanks. Thank you. Okay, a quick clinical correlate. I might not actually have you guys guess because I'm running slightly short on time, but here's the pica occlusion. I'll give you the answers. Lenticulostriate, this is just to show the different artery occlusions showing up on MRI can cause stroke and different, well, an occlusion in different arteries causes a stroke in different parts of the brain, glowing, shining brightly on MRI, depending on where the obstruction is happening. Okay, let's take a closer look at the main trigeminal sensory nucleus. So rostral to the spinal trigeminal nucleus is this main trigeminal sensory nucleus. It mediates touch sensation on the face and head and mechanosensation from the teeth. Neurons in this nucleus decussate and then ascend to the thalamic ventral posterior medial nucleus via the trigeminal lemniscus. We touched on this a little bit before, but just to reiterate. And then the mesencephalic trigeminal nucleus is a separate nucleus. It's located in the lateral portion of the periventricular and periaqueductal gray matter here in blue. These contain the cell bodies of muscle spindle sensory receptors that innervate the jaw. And these fibers travel to this nucleus via the mesencephalic trigeminal tract, a nice name that matches from tract to nucleus. And these are the jaw proprioception fibers. A third one, the caudal solitary nucleus. Ascending projections are focused on the parabrachial nucleus. I might have mislabeled the title of this slide, sorry. One ascending projection of the parabrachial nucleus is to the parvocellular division of the ventral posterior medial nucleus of the thalamus, and these help mediate visceral pain. So this is the parabrachial nucleus going up to a particular part of the thalamus, mediating visceral pain. Quickly, I wanted to mention the jaw jerk reflex. From the mesencephalic trigeminal nucleus, here in green. Neurons project to terminate in various brainstem sites important for jaw muscle control and proprioception. Again, mesencephalic trigeminal nucleus are receiving those proprioceptive fibers. In the jaw jerk or masseter reflex, a jaw spindle fiber innervated by mesencephalic trigeminal nucleus fibers, synapses with both the trigeminal main sensory nucleus in blue and the trigeminal motor nucleus in red. So it's hard to see, but this one neuron, it kind of bends around, connects and synapses with both of these trigeminal motor and sensory nuclei. And that neuron travels along to the jaw, to the masseter muscle, causing it to contract. Two thalamic nuclei that I've been mentioning, and now we're going to get a little more about. The ventral posterior medial nucleus, it processes mechanosensory information from the head, and then this information is projected to the lateral post-central gyrus, forming the face and head cortical sensory representations, which is the homunculus, which we'll see on the next slide. And then the medial dorsal nucleus. It receives information about pain, temperature, and itch. It receives trigeminal thalamic and spinothalamic inputs, and then it projects to the anterior cingulate cortex, which is important, interestingly, in the emotional aspects of pain, temperature, and itch. Itch, for some reason, I'm thinking of a lot because I get very emotional about my mosquito bites. Okay. Now the homunculus. These are my last two slides. We saw in the previous slide that projections from the ventral posterior medial nucleus form the cortical representations of the face and head. So coming from the ventral posterior medial nucleus of the thalamus, that's the head and face. And similar to the overrepresentation of the hands, the representations of the tongue and perioral region in the primary somatic sensory cortex are larger than that of other body parts because they're used more and used more extensively, particularly in speech and chewing, which humans do a lot of. In many species of rodents and carnivores, by contrast, the face representation is more extensive than that of the fingers and the tongue and the perioral region because of the large whiskers these species have, and the whiskers are their principal tactile, discriminative, and exploratory organs. And then from these sensory cortical representations, the primary somatic sensory cortex projects to higher order somatic sensory areas in the parietal and insular cortex for further and higher level critical elaboration of these sensory messages. Okay. Oh, this is just, this figure is another depiction of the homunculus, and I like it because it not only has the sensory homunculus that we saw before, but also the motor homunculus, which is similar but not completely the same. And then I also like it because it has the principal vasculature, principal artery that supplies that region, and you can correlate deficits in a person, perhaps to which artery is affected. So that concludes the content portion. Thank you guys for listening. I can take some questions. I think we won't do the practice questions, but if there are any questions, I'll take those at this time about the content.
Yeah, this is a hard chapter. You did a great job, Calvin. I've been really impressed with everything that everybody's really done. All of you guys have really impressed me. You know, I'll talk to, I think Jake is up next week. I'll talk to him to see if he's going to be, he's in the middle of orientation, so we may need to go an additional week before we can do his chapter. How are you guys feeling? I don't know what everyone's summer schedule is like. I don't know if rotations are starting up and people are unable to do, make their dates. Just kind of want to gauge how things are going.
Hey Shane, it's Keith.
Hi.
Hey, yeah, these have been great, Shane. I know for myself, I think I'm starting up rotations in about 2 weeks. I think Jeff already started.
Okay.
So I think I did. I think I finished the talks that I was signed up for, but
Yeah.
I guess I won't be able to make it for these sessions in about 2 weeks.
That's fine. I've been, I just kind of got on my research, my, off service time. So I'm kind of getting caught up. So I'll be updating the website. All this stuff is going to be going up shortly, and everybody's Anki Denks will be put up there after I've been able to sort of review them for content. So all that stuff will be there. So if you can't make it, it's not a big deal. You can always reference these things later on. But it
Perfect.
sounds like the majority of the group is okay with continuing. So I think that we can at least try. And if there's any issues, obviously, we'll figure it out as any problems come up. Okay.
Excellent.
Sounds good to me.
Thanks so much, Calvin. You did a great job.
Thanks.
All
I
right.
enjoyed it. Cool.
Thanks, everybody.
Alrighty.
All right. I'll send out an email. Hopefully I see you all next week. Bye.
Bye.
Thank you.